Hello everyone, welcome to the SWAYAM course of Genetic Engineering and its recent advancement. I am Dr. Richa Sahu, working as an assistant professor at MIT Institute of Biotechnology, MIT University, Chhattisgarh. Today we will be talking about regulation of gene expression. So, in the previous lectures we have talked and discussed about how the DNA is organized into genes, how genes store genetic information, how they replicate, how this information is expressed through the process of transcription and translation, that means how the genes are expressed. So, now let us talk about the most fundamental questions in molecular genetics, that is how the genetic expression is regulated. Most the bacterial gene products are present continuously at the basal level, that means few copies in the genome. The concentration of these products can increase dramatically when required. That means when the bacteria is in the environmental condition or in a stress condition, when it requires those particular genetic products or proteins, they are synthesized in multiple amounts. So, clearly fundamental regulatory mechanism must exist to control the expression of the genetic information. Bacteria or prokaryotic genome have been specially useful research organism in genetics for a number of reasons. For one thing, they have extremely short reproductive cycles and literally hundreds of generations, giving rise to billions of genetically identical bacteria in overnight cultures. The regulation of gene expression has been extensively studied in bacterial and prokaryotic genome, especially in Escherichia coli. Highly efficient genetic mechanisms have evolved in these organisms like bacteria and E. coli to turn transcription of specific genes on and off, depending on the cell's metabolic need for the respective gene product. Not only do bacteria respond to changes in their environment, but they also regulate gene activity associated with the variety of cellular activities, including the DNA replication, recombination and repair of DNA with cell division. During 1900s or early 1900s, it was shown or it was experimented that when lactose, let us for example lactose is present in the growth medium of yeast or Saccharomyces cerevisiae, the organism synthesizes enzyme that is required for the lactose metabolism. When the lactose is absent, synthesis of these enzymes diminishes at the basal level. That means if in an environment or in a media where the bacteria is growing, any kind of substrate is present that has to be metabolized, the bacterial genome automatically synthesizes the machinery or the enzyme required for the metabolism of that particular substrate. Soon thereafter, investigators generalized the bacteria adapt to the environment, producing certain enzymes only when specific chemical substrates are present. These are now referred as inducible enzymes. To contrast to that, enzymes that are produced continuously regardless of the chemical makeup of the environment are known as constitutive enzymes. That means they are mostly present at the residual level, they are always present irrespective of the environment the bacteria is in. Studies have revealed a contrasting system, however, whereby the presence of specific molecules that inhibits the gene expression. For example, tryptophan. Tryptophan plays a very important role in repressing the transcription of mRNA, which is needed to produce tryptophan synthesizing enzymes. That means, if tryptophan is present in the medium, in contrast to the inducible system controlling lactose metabolism, the system governing tryptophan expression is said to be repressible. That means, if tryptophan is present in the medium, automatically the cell stops synthesizing the machinery which is required to synthesize tryptophan inside the bacteria. Regulation, whether of the inducible or repressible type, may be under either negative or positive control. Under negative control or negative gene expression control, I should say, genetic expression occurs unless it is shut off by some form of regulatory molecule. In contrast, under positive control, transcription occurs only if the regulatory molecule directly stimulates the RNA production. In bacteria, genes that code for enzymes with related function tend to be organized in clusters on the bacterial chromosome, and transcription of These genes which are clustered together are often under the coordinated control of a single regulatory region. Such clusters of genes, including their adjacent regulatory sequences, are known as operons. So, the concept of operon model or operons, it was introduced in around 1960 by Jacob and Monod. They proposed that a hypothetical mechanism involving negative control that may be called the operon model, in which a group of gene or a cluster of gene is regulated and expressed together as a unit. The gene in an operon system or operons are transcribed together as a single mRNA molecule. This allows the gene to be controlled as a unit. Either all are expressed or none are expressed. Each operon needs only one regulatory region, including a promoter region where RNA polymerase binds, an operator region where the regulatory protein bind. In prokaryotic cells, there are three types of regulatory molecules that can affect the expression of the operons. First is the activators. These are the protein that increase the transcription of the gene, thereby increasing the translation of that particular gene or the proteins. Next is repressors. These are the proteins that suppresses transcription of the gene. Finally, inducers. These are the molecules that bind to the repressors and inactivate them. You can see the image of operon here. So, as I said, there is a cluster of gene. Gene 1, gene 2, gene 3 are the clustered gene. Before that, you can see a promoter region and an operator region. These three genes are transcribed as a single unit or a single mRNA, and this single mRNA for these three genes is translated into three different proteins. This is a generalized image of an operon system. The regulatory DNA sequences, in addition to the promoter, are the binding sites for the regulatory protein that turn the expression of the operon up or down. So, as I told you, there are three types of regulatory molecules or regulatory protein. Let us talk about repressors first. In this image, you can see how the repressor works. The regulatory protein repressors bind to the DNA sequence or the piece of the DNA called as operator. The operator region is present in the promoter area where the RNA polymerase bind. So, if the repressor binds to the operator regions, the repressor reduces the transcription. How? By blocking the binding of RNA polymerase on the promoter region, so that it can move forward to initiate the transcription. Or if on the operator region repressor molecules is bound, RNA polymerase cannot bind on it, so that the transcription is blocked. So, in this image the same mechanism has been shown. O stands for the operator region and P stands for the promoter region. The repressor molecule binds to the operator region, so that the RNA polymerase cannot bind to it, to the promoter region. If RNA polymerase cannot bound to the promoter region, the transcription cannot be initiated. So, the transcription and finally the translation is blocked, no protein is synthesized. Next is the activator. Some regulatory protein known as activator, they bound to the DNA binding region which is present just before the promoter. It increases the transcription of the operon by helping the RNA polymerase to bind the promoter. If the activator is present or bind to the DNA binding region or activator region, it helps the RNA polymerase to bind to the promoter region and it lifts up or it increases the transcription, so that a lot of mRNA is produced and the protein synthesis is also enhanced. Next is, there are some regulatory proteins, they can themselves be turned on or turned off by specific small molecules. What are these small molecules? These small molecules bind to the protein, changing its shape or conformation, altering its ability to bind to the DNA or the DNA sequence. For instance, an activator may only become active or can able to bind to the DNA sequence when these small molecules are attached to it. You can see in the image there is activator which is present, there is a DNA binding site, but the activator is not able to bind to the DNA binding site. If these small molecules, as you can see the small molecules, if they bind to the activator, the conformation of the activator changes and it can now bind to the DNA binding site. So, these were the regulatory proteins. So, now there are two types of gene regulation, positive gene regulation and negative gene regulation. What is positive gene regulation? Positive gene regulation is the type of gene regulation that enables the expression of genes. Negative gene regulation is the type of gene regulation that prevents the expression of gene. So, what is the difference, the basic difference between these two? In case of positive gene regulation, the regulation in which the presence of certain regulatory factors promotes the quantitative expression of genetic information. However, in negative gene regulation, the existence of certain regulatory components that reduce the expression of genetic information. Positive gene regulation is the form of gene regulation that allows gene to be expressed. On the other hand, negative gene regulation is the form of gene control or gene regulation that aids to the prevention of gene expression. In positive gene regulation, transcription factors bind to the promoter region of the gene. In negative gene regulation, the repressor bind to the operator region of the gene. In positive gene regulation, the binding of the transcription factors allows RNA polymerase to bind to the promoter region. On the other hand, in negative gene regulation, the binding of the repressor to the gene operator region prevents the RNA polymerase to bind to the promoter region, hence blocking the transcription. The example of positive gene regulation is tryptophan operon, and a negative gene regulation is lac operon or lactose operon. We will be talking about lactose operon in detail in forthcoming slides. So, let us talk about negative and positive regulation, how the mechanism works. On the left hand side, you can see the negative regulation. In negative regulation, the regulatory protein or the regulatory signaling molecule bind or inhibits the transcription process. So, in the presence of the repressor molecule, the transcription is blocked. Okay. So, in the image you can see that there is a promoter region, there is a operator region. In the operator region, the repressor is bound, because in the repressor is in the operator region, the RNA polymerase cannot bind to the promoter region and the transcription is blocked. So, the removal of repressor from the operator region in a negative regulation can take place in two ways. You can see the small regulatory or the signaling molecule. One way is the signaling molecule can bind to the repressor, change the conformation of the repressor and can unbound the repressor from the operator region. It can remove it from the operator region so that the RNA polymerase can now bound to the promoter region and initiate the transcription process. Or that the signaling molecule is bound to the repressor region, then only it can bound to the operator region. Once the signaling molecule is removed from the repressor, you can see there is a conformation change in the repressor molecule and it leaves the operator region. And once it leaves the operator region, the RNA polymerase binds to the promoter region and initiates the transcription. In case of positive regulation, the bound or the binding of activator initiates or brings up the transcription process. You can see there is the promoter region, and just before the promoter region there is a binding DNA binding site where the activator is bind. The activator initiates or increases the transcription process. How? There is a signaling molecule. If there is two ways again. If the signaling molecules binds to the activator, it gets removed from the DNA binding site. The transcription again slows down. Or another way, the signaling molecules binds to the activator, then only it binds to the DNA binding sites and helps the RNA polymerase to initiate the transcription process. This is how the positive regulation of gene expression works. Now, talking about the types of operons, there are two types of operons. First is the inducible operons, another one is the repressible operons. Inducible operons are the type of operons which is switched on in the presence of an inducer. The example is lactose operon. The repressible operon, on the other hand, it is usually present in the anabolic pathway, it is usually on. The operon is active and the functional product or enzyme is present normally in the cell, it is normally on. When the end product reaches the maximum value, operon is turned off. The example of a repressible operon is a tryptophan operon. Let us talk about lac operon, which is a type of inducible operon. So, in a lac operon there are structural genes, is the cluster gene. There are 3 types of gene in a structural gene. Gene coding for the primary structure of an enzyme are called as structural gene. There are 3 structural gene in the lac operon. The first one is the LAC-Z gene that encodes for the enzyme beta galactosidase. It is an enzyme whose primary role is to convert the disaccharide lactose to the monosaccharide glucose and galactose. This conversion is essential if lactose is to serve as the primary energy source in the glycolysis. The second gene that is present in the lac operon is LAC-Y, specifies the primary structure of permease. This enzyme permease facilitates the entry of lactose into the bacterial cell. The third gene LAC-A codes for the enzyme transacetylase. All 3 genes are transcribed as a single unit, resulting in a polycistronic mRNA. This results in the coordinate regulation of all 3 genes, since single messenger RNA is simultaneously translated into all 3 gene products or proteins. You can see the image of lac operon here. The yellow region is the repressor gene. Just after that is the promoter region and the operator region, which is the regulatory part of the lac operon. After that there are the 3 structural genes, LAC-Z, LAC-Y and LAC-A. You can see the after translation a single mRNA is produced as a polycistronic mRNA, and they are then translated into 3 different proteins: beta galactosidase, permease and transacetylase. How the lactose or the lac operon works? If the lactose is absent, let us say if the lactose is absent, this operon is switched off. How it is switched off? There is a repressor molecule or repressor protein is synthesized. It binds to the operator region. When it binds to the operator region, the RNA polymerase cannot bind to the promoter region. Transcription of the 3 genes into a polycistronic mRNA stops. It blocked, and so the synthesis of the all 3 enzymes is stopped. Why is it that? Because if the lactose is not present in the medium, you do not need the machinery which is used to metabolize the lactose. So, what if the lactose is present in the medium? In the cell, inside the cell, when outside the lactose molecules are present, some amount of permease, some amount of beta galactosidase and some amount of transacetylase is present in the cell already, even if the lac operon is switched off. So, when the lactose is there in the medium, that permease enzyme allows that lactose to enter into the cell. But when the lactose enters the cell, it will not be converted into galactose and glucose by beta galactosidase. This beta galactosidase, however, changes this lactose into allolactose, which is an isomer of lactose. This allolactose is actually the inducer of lac operon. It binds to the repressor protein, which was initially bind to the operator region. When this allolactose binds to the repressor protein, it changes the conformation of the 3D structure of the repressor. It unbinds from the operator region. When it removes from the operator region, RNA polymerase automatically binds to the promoter region and initiates the transcription of all the 3 genes into a single polycistronic mRNA, and the synthesis of all these 3 protein increases to 10 raised to power 3 folds. So, as I told, in the absence of lactose or the absence of allolactose, I should say, the repressor protein binds to the operator region, blocks the RNA polymerase from transcribing the structural genes. This is called as, in this condition, the operon is called as switched off. However, in the presence of lactose, when the cells are provided with lactose, the lac operon is induced. The inducer in the lac operon, as I told, is not lactose but allolactose, an isomer of lactose. After the entry into the cell, the few molecules that are present in the permease, it lets the lactose to come inside the cell. The lactose, as I told in the image, as I was explaining in the image, the beta galactosidase converts the lactose into allolactose molecules. This allolactose molecule binds to the repressor, which results in the release of the repressor from the operator. This triggers the repressor to get unbind from the operator region, and the RNA polymerase binds to the promoter region, which allows the expression of lac operon gene, and the amount of protein, that is all the 3 proteins, increases multiple folds inside the cell, so that the lactose can now be metabolized. The next is in lac operon is catabolite repression. It is also known as glucose effect. It is one of the very important phenomena that has to be understood to completely understand the concept of lac operons or any kind of operon. If you have cooked food in front of you, served, you will not go to the kitchen and cook food again. Obviously, you will save the energy because you already have a cooked food. Similarly, if in the media glucose is present along with galactose, the bacteria or the cell will tend to take the simpler sugar and try to metabolize it. Rather than taking a disaccharide, it will prefer the monosaccharide. But in case of lac operon, this phenomena where the glucose is taken by the cell in place of lactose is known as, this results in a phenomena known as catabolite repression or glucose effect. The enzyme for lactose utilization can be induced by adding lactose in the growth media, but it would be wasteful to induce these enzymes if the cells are already growing in a carbon source which they could use more efficiently than lactose. That is, if the glucose is present in the medium, it will be utilized more efficiently by the cell. This regulation is governed by a phenomena called as catabolite repression, also known as glucose effect. Catabolite repression ensures that the cells are using the best carbon source first rather than using the complex one. In catabolite repression, the binding of RNA polymerase to promoter region is regulated by cyclic AMP and CAP protein. CAP is catabolite activator protein. This catabolite activator protein or CRP protein is an allosteric protein which binds to the DNA binding site only if it is first bound by cyclic AMP. That means there has to be an association of binding of cyclic AMP with CAP. Then only CAP can bind to the CRP or DNA binding region. Cyclic AMP is synthesized from ATP by an enzyme called as adenylase cyclase, and glucose molecules inhibit the synthesis of cyclic AMP and stimulates its transport out of the cell. So, what happens now if the glucose is high? If glucose is present in the media or the glucose is high in the media, automatically cyclic AMP would be reduced. So, in the image we can see that if there is low glucose molecule, that means there would be high cyclic AMP. There is a CRP site, CRP binding site, it is a DNA binding region. In the CRP site, the CRP protein or the CAP protein binds, but it can only bind to that area when it is bound with cyclic AMP. So, there should be an association with CAP or CRP with cyclic AMP, so that together they can bound to the CRP site. When they bound to the CRP site, they facilitate RNA polymerase to bind to the promoter region and they start the transcription. But in case of high glucose level, if the glucose is present in the medium, automatically there would be low cyclic AMP. If the cyclic AMP is less or if it is not present inside the cell, the CRP protein cannot bind to it. If the CRP protein cannot bind to cyclic AMP, it will not be able to bind to the CRP site or the DNA binding region. So, the transcription will not be blocked, but it will be slowed down. So, transcription will be initiated, RNA polymerase will bind to the promoter region, but the overall transcription rate will be reduced. So, as I told you, when the level of glucose in the environment is low or nil, abundant cyclic AMP binds to the CAP to form the cyclic AMP and CAP complex. This complex binds to the CRP site. The efficiency of RNA polymerase binding is increased in the lac operon promoter region, resulting in the higher level of transcription of the structural genes. However, when the glucose level are high, no cyclic AMP is made. So, there would be no cyclic AMP CRP complex. If this complex is not formed, the CRP protein or the CAP protein cannot bind to the DNA binding region, and the RNA polymerase cannot bind to the promoter region efficiently. It will bind, but not very efficiently. In this case, the transcription occurs at a very low level. The similar phenomena, this phenomena is properly explained in this image, where you can see if the cyclic AMP is present with CRP protein, they complex together, they bind to the CRP site and initiate the transcription at higher level, because they help the RNA polymerase to bind to the promoter region efficiently, so that the structural genes are transcribed at a higher level. In the presence of glucose, the cyclic AMP reduces or becomes nil, which results in the cap protein will not bind to the CRP site, because there would be no cyclic AMP and CRP complex made. The RNA polymerase will bind to the promoter region, but not very efficiently. That is why the transcription will not be blocked, but it will be diminished. The level of the transcription would be low. So, this was about lac operon, where I explained the regulation of the gene expression in the prokaryote, taking example of lactose operon. There are some more operon systems that are present in prokaryote, that tryptophan operon, which is an example of repressor or repressible operon. So, in this lecture we talked about what are the regulatory mechanism of the gene regulation in prokaryotes, what are the type of regulation that we studied about negative regulation and positive regulation. We studied about the types of operon, and taking example of lactose operon, we studied how in the presence and absence of lactose this operon system works. We also talked about what if a simple monosaccharide like glucose, a simple sugar is present in the medium, what will happen. We talked about catabolite repression in this section. So, let us talk about how the lac operon works in the presence and absence of lactose. As it is an example of inducible operon, this operon is normally switched off in the presence of an inducer. So, what if the lactose is absent in the media? If the lactose is absent in the media, the repressor molecule binds to the operator region of the operon. If it is bound to the operator region of the operon, the RNA polymerase cannot bind to the promoter region, which results in the blockage of the transcription of the structural genes. So, now what happens if lactose is present in the medium? Even if the lac operon in the absence of lactose is switched off, the three enzymes, that is trans-acetylase, permease and beta galactosidase, they are present in a very small quantity inside the cell. If the lactose is present in the medium, this permease enzyme allows the lactose to get into the cell. When the lactose enters the cell, the enzyme, the residual beta galactosidase enzyme converts lactose into allolactose. So, the inducer, as we can say, the inducer of lac operon is not lactose, but allolactose, which is an isomer of lactose. This allolactose now will bind to the repressor molecule, and the repressor molecule is sitting on the operator region. The allolactose, when binds to the repressor molecule, the conformation of the repressor molecule changes, which results in the removal of the repressor molecule from the operator region. Now, when the repressor is removed, the RNA polymerase binds to the promoter region quickly and initiates the transcription of structural genes. Now, as the repressor has been removed and lactose is present in the medium, the transcription of the structural gene increases many folds, and large amount of all the three proteins, that is trans-acetylase, permease and beta galactosidase, starts translating into the cell, so that the lactose that is present in the medium can be very well metabolized. This is how the lac operon works in the absence and presence of lactose molecule. This completes regulation of gene expression in prokaryotes. Thank you very much. We will meet again.